I think this is the most important video in terms of being able to understand the whole receptors and the function in the organs. So, when we talk about preganglionic neurotransmitter, we're only talking about acetylcholine because there is no preganglionic adrenergic receptors. They're not muscarinic. They're not adrenergic. They're all acetylcholine. So, acetylcholine binds the nicotinic acetylcholine receptors in the preganglionic neuron. This results in influx of calcium and sodium, so you get propagation of action potential from the preganglionic neuron to the postganglionic neuron. So, cholinergic receptors, two subtypes, nicotinic and muscarinic. Nicotinic receptors are present in the nervous system. This is part of the receptor pathology present in Alzheimer's disease. So skeletal muscle, the acetylcholine receptor is nicotinic there. Also the ganglia. The ganglia's acetylcholine receptor are nicotinic. So when you activate them again, you get influx of sodium and calcium, and it's stimulatory. Now, cholinergic receptors that are muscarinic are either M1, 3, and 5, or M2 and 4. The 1, 3, and 5 work via the GQ receptors, so they result in activation of IP3, DAG, and result increase in intracellular calcium and also protein modulation, which results in stimulatory effect. M2 and 4, though, they activate GI, so they decrease the CAMP concentration and therefore are inhibitory. So when we move on to the adrenergic receptor, now we're only talking about the postganglionic receptors. They can be alpha 1, 2, or beta receptors that are beta 1, 2, 3. There are multiple subtype categorization, but the super subtypes are not as important. So when you stimulate the alpha 1 receptor, it acts via the GQ, increased concentration of IP3 and DAG, increased calcium concentration intracellularly, so alpha 1 stimulation should be stimulatory. Alpha 2 receptor, on the other hand, works via the inhibitory G protein. So you get decreased AMP concentration, so inhibitory. Beta receptor, all the subtypes, they work via GS. So stimulatory G protein increases production of CAMP, therefore they're stimulatory. So what does it all mean? So when you get sympathetic stimulation of the presynaptic postganglionic neuron, so again, this is the neuron that gets that synapses with the end organ. So when you activate alpha 2 presynaptic receptors, the effect is inhibitory. When you activate the beta 2 presynaptic receptor, the response is stimulatory. So again, that's the postganglionic neuron. So sympathetic postganglionic postsynaptic neuron, that's this one. You have alpha 1 receptor, that activation of it results in stimulatory response. Alpha 2 would be inhibitory response, and beta receptors, it would be stimulatory response. Now, what about parasympathetic system? So in the parasympathetic system, in the postganglionic neuron, when you get M1, 3, and 5 activation, you get stimulation. M2 and 4, you get inhibition. So we said when we activate beta 2 receptor, the result is stimulatory, and in case of a smooth muscle in the bronchial tree, when you stimulate them, you should get constriction, right? So why would you get bronchodilation when you activate the beta 2 receptor? This actually took quite a while for people to figure out. So when you get activation of the beta 2 receptor in the bronchial smooth muscle, it results in phosphorylation of the muscle regulatory proteins that lead to bronchodilation. Same in bronchial tree as vascular tree. When you activate the beta 2 in the smooth muscle of the vascular tree, you get vasodilation. So this is the slide that is highly board relevant, and not the slide, sorry. This is the video that's highly board relevant. So I think this would be beneficial for you guys to look at this a couple of times if you have any questions. And I'll be happy to answer any questions that you bring to the actual lecture.